and for putting this together in this wonderful meeting and giving me the opportunity to present. So my lab focuses on computational modeling of genome organization. And the reason we are interested in the genome is because it contains a complete set of DNA. And I suppose instruct the development and operation of an entire organism. And because of its importance, the genome has often been poetically called the blueprint of life. Now, since its discovery as the genetic material, significant effort has been devoted to study the DNA. And an obvious question is, how does the information transfer occur, right? How do you go from base pair to a walking and talking person? Now, a lot of progress has been made in that regard. And one of the important findings is that the genome structure is actually quite complicated. So one cannot simply view the genome as just a very long double-stranded helix, at least not for human. So instead, the genome is actually packaged in a very exquisite way to both fit inside of the nucleus and to enable gene regulation. So as shown here, the human genome actually consists of more than 6 billion base pairs. That's about 2 meters long when fully extended. And this set of molecules is now confined into a nucleus of only about 10 micrometers. So there's this 5 orders of magnitude difference in terms of the length scale. And this length scale difference really sort of necessitates genome packaging. And as shown here, the genome is kind of packaged in the genome packaging occurs over multiple length scales in kind of a hierarchical manner. And remarkably, cells have also evolved ways to essentially take advantage of this packaging kind of at every step along the way. And for example, the formation of the nucleosome essentially precludes other proteins from accessing the DNA. So here the DNA is showing gold here. In this sort of closed conformation, there's a tight bond to the histone proteins. So then essentially by regulating this population of the closed and open conformation, you can sort of regulate the DNA accessibility and in turn gene expression. Sort of a similar argument can be made for a strand of nucleosome. Here you have open and closed chromatin, which can again sort of regulate DNA accessibility and gene expression. And now at this larger scale, over millions of base pairs, the formation of so-called chromatin loops, they can again bring DNA segments that are kind of far apart in sequence now into spatial proximity. And this sort of 3D contact further allows cells to encode a complex gene regulation. So in some sense, I like to think of the genome really kind of like an enzyme instead of a long linear polymer. It really has to fold in 3D to form active sites to catalyze chemical reaction. And in this case, the reaction is, of course, gene transcription. Now, in addition to its biological significance, the reason that we are particularly interested in the genome is also because it's actually a very fun and a complex problem. And there's a lot of sort of fascinating phenomena that's occurring. And better yet, much regarding sort of how the genome sort of is established in 3D, the underlying mechanism remains unknown. So there's a lot of opportunities for discovery. Now, of course, sort of opportunity always comes with the challenges. And the first challenge is, is that the genome is very large, right? As I mentioned, 6 billion base pair, that's a lot of atoms. And just the sheer size of the system actually creates a lot of sort of issues. And we as computational chemists over the years have become very good at modeling sort of a small scale system, but for this kind of larger scale system, there's still a lot of difficulty. And the second challenge is that the genome is really, really large. I couldn't emphasize that enough because 2 meter long, that's a really macroscopic scale molecule. And so because of this complexity of genome organization, we try to study the genome sort of at different scales with different approaches. On the one hand, we try to study the whole genome organization. There we use an integrative approach that combines modeling with experimental data. And on the other hand, we also try to study the genome at a smaller scale with the atomistic details. And today I will focus on our whole genome modeling effort. So there, we are particularly interested in the principle of genome organization, in the mechanism that establishes the genome structure in 3D. So as you might expect, the genome is very complex, and so is its mechanism, right? So today we're going to focus on one particular aspect that has gained a lot of momentum recently. Now, before I dive into details, I want to remind you that in the nucleus, in addition to DNA, as shown here, there are also these membraneless structures called nuclear bodies. They include nucleolus and speckles. And so there is evidence that both the genome and the nuclear body, they use the liquid-liquid phase separation for their organization and for their formation. And for example, EM image of the eukaryotic genome had a long review that there are two types of chromatin that sort of occupy spatially distinct regions. There is euchromatin in bright and heterochromatin in dark colors. And this organization is kind of reminiscent or consistent with a picture of block-o polymer phase separation as shown here. Now, the liquid property of nuclear bodies include their round morphology and their fusion upon contact. So this is basically a time trace of two contacting nucleoli that eventually sort of fuse into a larger droplet. And this is kind of a typical phenomena of liquid droplets. Now, phase separation is a very intuitive idea, and it has it's consistent with a wide variety of experimental observations. These are just two examples. However, I want to emphasize that the phase separation also cannot explain many other experiments. In particular, recently, several groups have found that certain genomic loci actually have very well defined distance to nuclear speckles. And this kind of very precise distance is actually inconsistent with a liquid picture. If we think of the nucleus as just a liquid, then by definition it's supposed to be very dynamic and homogeneous, which is really cannot encode very precise distances. So another puzzling observation is the apparent coexistence of multiple droplets. So here the cells are stained with antibody to basically highlight the number of nucleoli, and this is a quantification of the image shown on the left. So here you clearly see multiple nucleoli. And why is this interesting? It is interesting because it seems to be inconsistent with this classical nucleation theory, as you might know. And so this theory tells you that the stability or the free energy of a droplet can depend on two terms. One term comes from the surface tension, sigma here, and is proportional to the surface area. And then the other term that arise from the interaction among particles and is proportional to the volume. And with this expression, you can show that the free energy will always decrease when the droplet fields merge with each other. So then basically the thermodynamic equilibrium really just consists of one single droplet as shown in this cartoon here. So then the obvious question is that what drives the inconsistency between experiment and the theory, and what is maybe so special about the nucleus environment that sort of stabilizes apparent multiple droplet state over the entire cell cycle. So these are the kind of questions that we would like to understand better with the computational modeling. But to study the genome organization and phase separation in the nucleus, we first need to build a model for the genome. But as I mentioned in the beginning, modeling the genome can actually be quite challenging just because of its sheer size. To basically make any meaningful progress, we essentially have to throw away our beloved atomistic detail and work with coarse grained models. So this is basically a cartoon for our 3D model. We have sort of each one of the 46 chromosomes. We represented them as a bead on a string, and the chromosomes are sort of confined in a sphere to mimic the nuclear envelope. So in one resolution that we work with, each chromosome is each bead in the chromosome is represents 1 million base pairs. And now with this coarse grained representation, you can define an energy function, basically sort of quantify the stability of different genome structures. So the design of the energy function itself is actually a very interesting topic, and basically through trial and error, you can sort of actually learn a lot about the mechanism of genome organization. But in particular, we show that a simple block of polymer phase separation actually cannot explain many features of the human genome organization. But I won't go into too much details. But now, once you have the energy function, right? Once you establish the functional form, you still need to sort of parameterize the interaction within the particles, because though the interaction really to quantify the energy and sort of dictate the final structure that you're going to get. Now, to be frank, coarse graining is actually a very popular technique in the theoretical chemistry community. However, we cannot just directly borrow existing techniques and try to parameterize the genome model. And the reason is that, as I mentioned, each one of our beads actually contains many atoms, and sort of just the sheer size and the complexity of the chemical composition of each bead basically renders the sort of traditional so-called bottom-up approach inapplicable. In particular, we cannot afford to just run quantum mechanical calculation or atom simulation to try to parameterize the interactions and parameterize the genome model. So basically, to parameterize the model, we really kind of have to think outside the box. And in the end, we established we call this an information theoretical approach. And what this allow us to do is basically extract parameters from experimental data. So the experiment that we use is called a Hi-C, and it's actually very similar to NMR, but for the genome. So what it does is basically it detects contact between the DNA segment in the nucleus with cross-linking, as sort of illustrated here. And the result is typically shown in as a contact map, and each data point in this contact map basically tells you the probability for a pair of DNA segments to be in contact in the nucleus. Now here, in this color scale, the probability decreases from yellow to red and to white. Okay, and this diagram basically illustrates how our sort of methodology works. We start with an energy function. And with this energy function, we can then basically simulate an ensemble of genome structures, as shown here. And from this ensemble of structures, we can then compute an in silico contact map. Then we can look at the difference between the simulation and experiment. And we can use that difference to basically update the parameter to improve the energy function. And we can do this iteratively, so that in the end, we arrive at a converted energy function and a converted genome structure that reproduces experimental data. So I mean, I won't go into detail, but we do have a nice little argument to say that the model we have is supposed to be optimal given experimental data. So after we obtain an ensemble of structures, we carried out extensive validations to ensure their biological relevance. So it's probably not too surprising that our model was able to reproduce Hi-C data, given that they were actually provided for model training. So here the experiment is shown in bottom and the simulation is shown at the top. And these are basically just zoom in of a different part of the contact map. And you can sort of appreciate that our model was able to reproduce the contact sort of across several orders of magnitude. And we do have a quantitative metrics to, in addition to this diagram, to sort of ensure that the simulation is indeed agree with the experiment very well. And importantly, in addition to the Hi-C, we also compared with a set of qualitative and quantitative image results. In particular, the chromosomes are known to form territories, and you can readily see that from our simulation structure as well. Here, each chromosome is assigned with a unique color, and this is basically a blow up of the simulation structure. And you can see that each chromosome is more or less sort of collapsed. There's very little intermingling among them. And now if we instead color the genome with different chromatin types, you can see that there is a clear phase separation between euchromatin, which is shown in red, and the heterochromatin shown in blue. And the heterochromatin also sort of localized at the periphery, which is consistent with the EM image that I showed previously. And finally, when we look at the radial position of an individual chromosome, you can see that the simulation values actually correlate very well with the experimental results. So again, I want to emphasize that none of these results were actually provided for model training. So that the fact that we're able to reproduce them it sort of provides confidence in both our modeling strategy and in the biological relevance of the structures. So now that we have a structure, you might wonder, what can we do with that, right? What additional value do they add to the genome? And so I would say that one advantage of having the structure is that they're actually very useful for building intuitions. In particular here, in a collaboration with Brad Bernstein, we looked at the genome organization in normal and tumor cells. So here, if you just look at the high-C data, you sort of see some differences, but for example, the contact in tumor is a little bit weaker, but it's not clear to conceptualize, what exactly does that mean in terms of the 3D structure. But I hope you can appreciate this larger scale change, from our simulations. In particular, unlike what you see in tumors, unlike what you see in normal cells, in tumor, the heterochromatin, they actually now move away from the envelope and sort of now kind of aggregate in the nuclear interior. So we were very excited about this prediction, and we thought we wrote the manuscript. And as you might expect, we encountered reviewer number 3, and who were not very impressed, and they wanted to see more validation for the simulation predictions. And fortunately, we have excellent collaborators, and they performed electron microscopy on these tumor and normal samples, and lo and behold, they found exactly the same as our simulation prediction. In particular, here the heterochromatin are again showing dark color. You can see that in normal cells they localize at the nuclear periphery, but in tumors, and you see that they indeed now sort of dissociated from the nuclear envelope and moved to the interior. So that was kind of a record moment for me that the simulation agreed very well with the experiment. All right, and now that we have a reliable model for the genome, we can actually also now begin to think about the phase separation in the nucleus. To do that, we introduced the additional corresponding particles, and this particle, they basically try to mimic the nucleolar proteins, which are sort of the key component of the nuclear body. And we basically make sure that the concentration, the size, and the interaction with the chromatin of this nuclear of this corresponding particles sort of they match with those of the nucleolar proteins. And so this particle, they sort of have attractive interaction with each other to promote phase separation. They also can sort of interact with the chromatin favorably. They interact with the specific region of chromatin favorably. Basically, after introducing this corresponding particles, we can essentially perform dynamical simulation of phase separation in the nucleus. So here, the protein particle, they are showing in orange, and the chromosomes are showing in gray. And I'm just going to play one movie that sort of is a typical simulation trajectory. And as you can see that as the simulation evolves, we started from a kind of a random distribution of the protein particles. As the time evolves, you see that the particles themselves they will aggregate into small clusters, and small clusters will coalesce into bigger clusters. And interestingly, in the end, the simulation actually settled into 2 droplets, not 1, but 2 droplets. And that's interesting because that's again sort of kind of inconsistent with the expectation of the classical nucleation theory. But it is consistent with the biological observations. And we can also monitor the number of clusters as a function of time. You can see that basically you have an initial burst of clusters at the beginning, but then that gradually coarsen, but and eventually settle down to 2 droplets. And we performed additional simulations and just to see whether this is statistically significant. And as you can see here, in all but one simulation, we actually always sort of arrive at multiple droplets in the end of the simulation. So now this will actually become quite interesting. Now, before we get too excited, we also performed additional sort of comparisons with the dynamical measurements from experiment to sort of see whether the dynamics of our simulation makes sense or not. So in particular, here we looked at the study from the Branguen group. So here they actually monitored the coarsening kinetics of droplets directly in the cell nucleus. It's actually a very cool experiment. And what they did, they monitored the size of the average size of the droplets as a function of time. And you can see that this size increase there's a Pavlov behavior of the size as a function of time, and this exponent is around 0.1. And this exponent itself is kind of peculiar because it's it deviated from the canonical value, which is around 1/3. But our simulation was actually able to reproduce that very nicely. And in this other experiment from the Weska lab, so they directly monitor the fusion kinetics of 2 droplets, and they sort of measure the neck radii of the fusion as a function of time. And you can see that this radii increase sort of followed a parallel. and the exponent is around 1/2, and that agree with our simulation as well. And as a final check, we also sort of went back to our tumors, and we determined we basically performed a dynamical simulation of phase separation in tumors. And here, everything was kept the same regarding the protein particles, the concentration, the interaction with the chromatin. Everything was kept the same. The only thing that we changed was the interaction between chromosomes. So here now the interaction was derived from tumor high C instead of the normal high C in our previous simulations. And interestingly, that change actually resulted in more nucleoli in tumors compared to normal samples, as you can see here. And that's interesting because tumors are actually known to have more nucleoli. So the fact that we were able to reproduce that trend, at least qualitatively, is quite exciting. So again, all of this qualitative and quantitative comparison that sort of support the biological relevance of our simulation. So now we can maybe go back to look at the simulation details and maybe learn something about the system. In particular, we want to understand why are we seeing multiple droplets in our simulation, right? And as a chemist, I like to sort of think of this from a reaction perspective. So we introduce a reaction coordinate, and this reaction coordinate basically measures the distance between the two droplets. So at a larger distance, the two droplets are sort of separated from each other, and at a small distance, they will fuse. And we then computed a free energy profile along this reaction coordinate. So what you see is that, interestingly, we saw a free energy barrier, and this barrier appears when the two droplets sort of begin to touch each other. This is again sort of inconsistent with what you might expect from the classical theory of phase separation. And then we sort of try to understand, why do we see a barrier here? And we performed a free energy decomposition. We found that the barrier is mostly arise from an entropic origin, and then we sort of scratched our head a little bit, and we thought that there could actually be a very intuitive explanation for this entropic barrier. And the reason is that the protein droplets they are essentially tethered to the surrounding chromatin network because of favorable protein-chromatin interaction. So then, as the droplets try to sort of fuse and they move close to each other, they will basically pull on the chromatin network. They will basically put more constraint on the chromatin network. And the chromatin network is made of polymers. I guess in a naive approximation, they are kind of like rubbers. And when you pull on the rubbers, there are going to be a sort of countering force. And this countering force and is known to have an entropic origin, and that will sort of give rise to this barrier. So this kind of reminded me of this cable crossover that I kind of enjoyed doing before. But this is more like overcoming gravity, not really entropy, but you get the idea. So our study on this nucleolide kind of support that the chromatin network could actually impact the formation of nuclear bodies. But in the meantime, you can expect that the nuclear body, they will also impact the chromatin network as well. And that point actually really become very clear in our latest study, where we try to now build a more complete picture for the nucleus. So here, in addition to the genome and the nucleoli, we also included the speckle and the nuclear lamina. Those are sort of key components of the nucleus. So in this model, in addition to reproducing the genome organization, we are now also able to reproduce the contact between the genome and the various nuclear landmarks. So this is supposed to provide a more accurate picture for the nucleus. But interestingly, now this model actually also help us to understand how would the precise distance that I mentioned in the beginning would arise in the nucleus. And the idea is that we think that the genome and the nuclear body, that sort of can form through a self-assembly process through sort of basically a co-phase separation mechanism. And as sort of shown here in this mechanism, the nuclear body, they will basically nucleate around the chromatin segments as sort of because of favorable interactions. And this nucleation essentially ensures their sort of a close contact with the individual cells. And because this nucleation also happens in all cells, so this will essentially ensure that the certain genomic region to always have a very well-defined distance with the nuclear speckles. And this explains the precise distance that have been shown in previous experiments. But in the meantime, because phase separation is non-specific, in different cells, you also they basically the nucleation could use a different set of chromatin segments. So there is a heterogeneity in the chromatin segments that are nucleating the nuclear body in different cells, and this heterogeneous context could sort of give rise to the heterogeneous chromosome positioning in individual nucleus as well. So this a simple co-phase separation mechanism can actually reconcile this, both this precise and heterogeneous feature of genome organization, which may sort of seem a little bit contradictory, at the first sight. Okay.
Just
I showed you one mechanism about the genome organization, but we are trying to sort of use that to try to predict genome organization and to see the impact on genome function. And I'm running out of time, so I want to thank my lab member who actually did the work. I was fortunate to work with an excellent group of people, and thank my funding, and thank you for your attention. Yep.
Okay, very exciting talk. So you see, there are a lot of questions. Let me just read a few of them. You already answered my question on this polymer constraint thing. Actually, this guy, Robin Busma asked you, by starting with the high-seed map, are you not introducing by hand the incomplete phase separation that you are wanting to explain?
So with the high-seed, yes, we do in sort of yes. I mean, high-seed in fact more or less tells you this phase separation. That is correct. So high-seed encodes that, but high-seed does not really tell you much about the nuclear body phase separation. I guess I don't know which part of the phase separation the Robin was referring to. Yes. I mean, the fact that we were able to reproduce this phase separation, that is totally from high-seed data.
I was referring particularly to the chromosome territories, because the high-C map in a way includes all the physics which leads to the chromosome territories.
That's fair. Yeah, you are absolutely correct in that. So yeah, because that's why you can sort of see that there are the very kind of strong contact in each block that actually correspond to individual chromosomes. But I would say this part is it's definitely not apparent from high-C, that the fact that we were able to reproduce the radial position of chromosomes. So I mean, it is in some sense still encoded in high-C, but I would say it's not obvious that a model just based on high-C would be able to reproduce that in terms of the radial position of chromosomes.
Thank you.
Yeah, thank you.
So yeah, there are some questions on the details of your simulation. One thing, Pinaki Swan asked you, do the number of droplets in simulation depend on the interaction strength between chromatin beads and the protein beads?
Yes, that's a great question. Absolutely. So we actually showed that. I didn't show that result here. We actually have that in our manuscript. So basically, as we increase the strength of the interaction, we actually see more droplets. And the idea that basically the droplets that form through nucleation around the chromatin, then they will sort of basically fuse. And at a stronger strength, it will make the nucleation easier. So initially you have more droplets in the beginning. And because the coarsening sort of kinetically arrest, if you nucleate more, you're going to end up with more droplets. Yeah, that's a great question.
And also probably also the protein concentration, right?
The protein concentration, yeah, that's true as well. Yeah, we sort of just try to sort of more or less fix the protein concentration based on the biological values, but correct. Yeah.
Yeah. Raphael asked you, how do you determine when to stop the simulation in the phase separation study? How does the number of droplets depends on simulation time? So I want to make sure it's whether it's metastable or not, right?
That's an excellent point. I mean, I guess you can see that. We can sort of more or less map our simulation time scale to sort of experimental time scale. We sort of run about several hours. But if you look at the free energy, I mean, you are correct that the single droplet state is still the thermodynamic minimum. So that if you run it infinitely long, you would probably in the end still end up in this single droplet state. That's why we call this sort of kinetic arrest. But sort of in our reasonable simulation time scales, if we can map to biological value on the order of maybe several hours a day, I mean this basically gets arrested in its multiple droplet state. We do not see sort of further coarsening anymore.
Yeah. So let me see. Probably last question is Jing Chen asks you: are the droplets associated with the heterochromatin and euchromatin domain respectively, or other distinct connected domains?
Sorry, could you say that again?
Are the droplets associated with the heterochromatin and euchromatin domains respectively, or other distinct connected domains? So what's the nature of the droplets? Are they connect to the associated with the heterochromatin, euchromatin, or there are some other features?
I see. So this droplet, so these are, we're trying to model the nucleolus. So the nucleolus, they are actually, so they are sort of, they are functioning sort of for this ribosomal RNA. So they actually sort of associate with very specific set of chromatin, in particular those sort of segment for transcribing the ribosomal RNA, but they also have sort of maybe more than those regions like called nucleolus associated domains. So yeah, so I mean those are typically heterochromatin. You are correct. Yeah, but it's not just for heterochromatin. They are also important for transcription as well.